Okay, welcome back for our fifth lecture on the Chapter 21 Nucleic Acid and Protein Synthesis content. This is another longer one, so if you've been enjoying those relatively short ones, hopefully you can bear with us for a bit of a longer one as we start to understand, instead of just the DNA that we've been focused on, some RNA concepts. In particular, this lecture will focus on RNA and transcription. So we've focused already on DNA and taking DNA and unwinding it and making new DNA strands. And so now we're ready to move out of DNA and into RNA. So there are different types of RNA. Things are a little more complicated than what we had with DNA. So RNA transmits information from DNA to make the protein. So the DNA codes all this information in sort of long-term storage, and then the RNA is going to make use of it to help make these proteins that our cells need. There's several types, and we can see here we have messenger RNA, which is denoted by a lowercase m before RNA, and that's what carries the genetic information from the DNA to the ribosomes, which are the sites of protein synthesis. We have transfer RNA, lowercase tRNA, which brings amino acids to the ribosome to make the protein. And then we have ribosomal RNA, lowercase r RNA, and that's the most abundant type of RNA, and it's combined with our proteins to form the ribosomes, which are part of the cell, as you learned way back in middle school. So here we look at table 21.4, which gives the types of RNA molecules and their percentage in humans. We mentioned that the ribosomal RNA is the most abundant, and it's quite abundant there at 80%, and it's our major component of the ribosomes, which are the site of protein synthesis. The messenger RNA is present in the smallest amount of these three, about 5% of the total RNA. And that's the one that carries the information for protein synthesis from the DNA, where it's originally coded for in a nice stable long-term storage type mechanism in the nucleus out to the ribosomes. And then finally the transfer RNA, which makes up the remaining 15% or so, brings the amino acids to the ribosomes for protein synthesis to make those polypeptides or polyamide bonds. So looking at that transfer RNA, the last one we discussed will be the first one we'll look at. It has a 3' end with the nucleotide sequence ACC, which is known as the acceptor stem. So we see there the 3' with that free hydroxy group has that ACC starting, and then it contains an anticodon where we see the UUU down at that one loop. So there we have an anticodon to look at the end opposite that 3' end. And we see some hydrogen bonding to have those complementary bases form the double-stranded sections of RNA. Okay, looking further at RNA and protein synthesis. Protein synthesis involves the processes of transcription, in which the messenger RNA is formed from a gene on a DNA strand, and then translation, in which the transfer RNA molecules convert the messenger RNA information into amino acids to build that protein. So lots of good information happening here, and lots of similar sounding things. Just like we had nucleosides and nucleotides, now we have transcription and translation. Just be careful that you understand the subtleties in those similar sounding words. All right, so let's go into transcription first, which involves the synthesis of mRNA. So a section of DNA containing the gene unwinds, and then one strand of DNA is copied starting at the initiation point, which has the sequence TATAA, and then an mRNA is synthesized using complementary base pairing, of course, with uracil replacing the thymines, that TATA sequence as it's known. We have the newly formed messenger RNA moves out of the nucleus to ribosomes in the cytoplasm. So there we go. That's how we get this information, which stays nice and stable there in the nucleus in the DNA. It does need to eventually get out to the ribosomes, because those are where we're going to actually make protein. So we've got to get that information. It's nice that we have it stored stable in that nucleus, but it's got to reach the cytoplasm for any protein synthesis to occur. Now we've already talked about DNA polymerase, so there's also RNA polymerase. And during transcription, the RNA polymerase moves along the DNA template in the 3' to 5' direction to synthesize that corresponding messenger RNA. The messenger RNA is then released at the termination site where it's gotten the information it needs to code for the protein that it's coding for. So there we see a cartoonish depiction of that process where we have that RNA polymerase helping to synthesize that mRNA from the place where the DNA has opened up through the termination site. Okay, so let's look at mRNA processing here, the exons and introns. So in eukaryotes, DNA contains exons that actually code for proteins, and then introns which do not code for the protein. So originally a pre-RNA is formed that includes the non-coding introns. Later, the non-coding introns get removed, and then finally the exons are joined to form the functional messenger RNA, which goes to the ribosomes with the information necessary for the synthesis of the protein, and the intron unnecessary information left out. So here you see a cartoon where that intron removal occurs from the pre-messenger RNA. So we get our gene that codes for the protein. Then we have transcription where we form that pre-mRNA. And then there's the processing where we chop off those introns that don't actually code for the proteins. And then finally the messenger RNA that's resulting is really just the joined exons, which actually do code for the proteins that are going to be synthesized. Now with transcription ultimately resulting in protein formation, we need to have some regulation. So the transcription process is regulated by a specific mRNA synthesized when the cell requires a particular protein. So enzyme induction, for instance, where a high level of a reactant induce the transcription process to provide the necessary enzymes for that reactant. So it's similar to when we talked about our enzymes and the regulation of enzymes. Here, if we've got a high level of a reactant, then it kick-starts the whole process to get the transcription process going to provide those enzymes that reactant needs in order to react. So within a gene, we have sections of DNA called operons, and those regulate the synthesis of these related proteins. Okay, so for a specific example, we can look at the lactose operon and repression system. So the lactose operon consists of a control site and structural genes that produce messenger RNA for lactose enzymes. When there is no lactose in the cell, a regulatory gene produces a repressor protein which prevents the synthesis of those lactose enzymes. The repressor effectively turns off the messenger RNA synthesis. Unfortunately, some of us lose the ability to turn that back on, and we become lactose intolerant. Most of us as youngsters, as infants anyway, relying on mother's milk or some sort of stimulant, have the ability to digest lactose. Many of us keep that into old age. Some of us lose it at some point between infancy and old age. And so here we see a cartoon there of the lactose operon and what it looks like when it's turned off. So as I mentioned, some people lose the ability to turn it back on, and therefore they have lactose intolerance, which results in an inability to digest lactose. Unfortunately, the bacteria still can do so, and you'll often get stomach aches and diarrhea and all that good stuff if you become lactose intolerant and then consume lactose. So assuming that the person retains the ability to make the enzymes to digest lactose, well then in that cell, the level of lactose induces the synthesis of the enzyme required for its metabolism. So having lactose present in the cell is what kickstarts the messenger RNAs to be formed to make the proteins that will form those enzymes. The lactose combines with the repressor and then removes it from the control site, and that allows the RNA polymerase to catalyze the synthesis of those enzymes by the genes in the operon as long as the lactose is present, and as I mentioned, as long as the person still has the ability to make the lactose enzymes in the first place. So here we see another cartoon, and this time the lactose operon gets turned on. So we have the repressor messenger RNA, the lactose inducer, and that causes now the repressor to become an inactive repressor, and so it's not stopping the messenger RNA from forming. So we see the messenger RNA is allowed to interact and form the beta galactoside, galactosidase, actually the protein enzyme that allows for the breakage of the lactose link. So there you have it, a lactose operon turned on. All right, so now that we've had a little bit of time to consider RNA and transcription, we'll end this lecture with a learning check, as we so often do. In this case, we're asked, what is the sequence of bases in messenger RNA produced from a section of the template strand of DNA that has the sequence of bases 3', C, T, A, G, G, 5'. So go ahead, make your guess. Hopefully it's more than a guess, right? An educated guess. And then start us back up if you need a moment to stop while you decide on your selection, and start back up to check your work. Good luck. Okay, so hopefully you selected number 2 here, and there were a couple things to look for, right? First of all, if we're at the 3' to 5' on the DNA, then we're going to go 5' to 3' on the RNA. And the other big thing to look out for is that any of your A's on the DNA, instead of pairing with T's as they would on DNA, they pair with U's for uracil here on the RNA. So our C's and G's, that was no problem. Our T goes to A, but our A's, instead of going to T, go to U's. So that's why 2 was the correct choice. So hopefully that's what you selected, and hopefully you know why. If not, if either you selected an incorrect choice or you're just totally lost and need some help, then please do reach out to me for help before moving on. Otherwise, we'll meet again in the next lecture.